All right, so let's get started. It's 8 p.m. and then welcome everyone to our today's Museum seminar. So first of all, I want to say, Happy New Year. It's just one day from the Chinese New Year. If the audience, no matter in China or US or Europe, Happy New Year. It's really my pleasure to welcome our today's speaker, Dr. Rong Fan from Yale, as our very last lunar year 2023 speaker on the platform. So I think a lot of us actually already know Rong's work. It really actually really refreshing work, actually very beautiful technology development. So Rong's lab actually, Rong actually currently is Harold Hawkinson Professor of Biomedical Engineering and Pathology in Yale University. Rong actually received finished his undergraduate study in University of Science and Technology of China. And then he received a PhD in chemistry from UC Berkeley. Complete his postdoc training at Caltech, and then joined the faculty in the Department of Biomedical Engineering at Yale in 2010. So from 2010, as I just mentioned, he started his independent research program as assistant professor in Yale, and then all the way to 2020 as a full professor. And then 2022, he was appointed as Harold Hawkinson Professor of Biomedical Engineering in Yale. So Rong's lab is really focused on the development and the deployment of single-cell and spatial multi-omics profiling technologies, often based on microfabricated devices to investigate pathogenesis and the therapeutic response of complex human disease, including cancer, autoimmunity, and the cardiovascular disease. In particular, his lab is interested in hematological malignancies, brain tumors, and systemic lupus. He's also interested in cellular immune function characterization and the application to cancer immune therapies. A microchip technology his lab developed for simultaneous measurement of 42 immune effector protein in single cells, which remain the highest multiplexing to date for a single cell protein secretion assay, has been commercialized and currently used by over 100 pharmaceutical company and medical center in US and around the world, which I think is really remarkable. Recently, his lab developed a novel NGS-based approach called DeepSeq for spatial transdome mapping, spatial high-plex protein mapping, and spatial epigenome mapping, which may find widespread applications in development biology, cancer research, neuroscience, and immunobiology. So I think I don't want to really go through all the achievements from Rong's lab, but with all these breaking through discoveries and the new technology development, he was awarded with numerous awards, which including the Rising Star Awards, as a fellow of the American Institute for Medical and Biological Engineering, and then member of Connecticut Academy of Science and Engineering, Yale Cancer Center class of 1961 Cancer Research Award, and last year he became fellow of the National Academy of Inventors. So I'm super excited to learn what you are going to talk today, Rong. Really appreciate you contribute to our platform. Please go ahead.
Yeah, thank you. Thank you, Chuan, for a very kind of introduction. And also thanks, Xiaolei. I think you guys organized this immuno talk that has been really a major highlight for the entire immunobiology community. So I'm very humbled to have this opportunity to speak to you guys and share with you what we do. I really think the main purpose here for me is to get your feedback, to get the comments feedback from from immunobiology experts. And so this is my disclaimer. And I want to just use one slide to highlight what was kind of my turning point in my career. And when I finished my PhD, I was looking for a postdoc, but I had nothing. I had no track record in biomedical research, but I know that's something I would like to at least spend a couple of years kind of exploring and kind of enjoy something I never had experience with, but potentially exciting. And I moved to Caltech working with Professor Jim Heath. And then the topic I know I need to work on is cancer system biology. And then I need to raise something about cancer. So this is one of the review articles written by Professor Joanna Joyce. And by reading this, I realized, okay, the tumor microenvironment is fascinating, all right, and extremely heterogeneous. You really need a single cell resolution tool to really understand all different cell types there. And number two, ideally, you can really resolve the different cell types and what they do in a spatial resolved kind of tissue context, right? And but that time, I think it was pretty much like an ice age in single cell analysis. And if you can grab one cell, count the copy, how many copies of that particular protein, that was actually a science paper published by one of my best friends, Bo Huang. But if you really want to take that type of devices or technologies to understand so tumor heterogeneity, which means you need to can assay thousands or tens of thousands of single cells. Each cell you look at so many different proteins to understand at a system level. That was totally impossible to do with that device. So this is what I learned. So from the first thing I learned at Caltech, it's kind of like ELISA, but just a kind of array format of ELISA. After I learned this, I said, okay, since you can do ELISA microarray or antibody microarray, that allows you to do multiplex measurement of proteins. But you would not think you can do multiplex protein measurement using a DNA microarray or antibody microarray for single cells. But I wasn't, very sure how challenging it is. I said, okay, why not? If we can miniaturize in a microarray, okay, that's what I proposed back to that time. So kind of miniaturize in a microarray and put in a 1 nanoliter microchamber. If you're doing the math, okay, if you have one cell per nanoliter, and with all culture cells in a petri dish, we know that's 1 million cells per mil. And if you're doing the math, 1 million per mil is actually equivalent to 1 cell per 1 nanoliter. So that means the proteins produced by this cell in the 1 nanoliter chamber should have more or less the same concentration as in your petri dish, right? And then the question is, can we miniaturize our DNA microarray to do that? Okay, long story short, okay, I don't want to talk too much. Eventually got it work. We were able to do kind of a dozen proteins from a single cell. And in the beginning, I was trying to look at single cell cytokine secretion, but later in the same technology can be tweaked a little bit to look at intracellular phosphoprotein omics. And that was really my first experience in single cell analysis in cancer biology, but somehow that directed my interest towards immunology, because when you look at single cell cytokine secretion, what matters most? Okay, immune cells. And then we got an amazing opportunity to collaborate with Tony Ribas. And that time, what Tony told me is, okay, he's a big fan of cancer immunotherapy, but back to 2006, 2008, people don't care about immunotherapy, all right? And what Tony said is, okay, that's kind of like you're throwing a glass jar from the top of the USA Medical School building. Most of glass jar are gonna break, but you still have to do that because some patients might benefit, right? So I was so fortunate to have the opportunity to work with Tony. Eventually, we applied the technology we developed to look at the engineered T cells. Back to that time, the T cell engineered T cells. But the same finding back to that time, so if the T cells are polyfunctional, that can produce many different immune effector cytokines, they are very potent. They give you a much durable response in patients. So after I moved to Yale, I decided to take the same device to look for my new collaborators at Yale to kind of look at immuno-oncology stuff. But I realized actually I like the device, but my clinical collaborators never liked this very nice looking device. And this becomes an engineering question. Can you really make the device much higher throughput, much easier to use, get rid of those spaghetti stuff, make it much more scalable, manufacturable? So since I'm in School of Engineering, I said, okay, I'm going to take on this engineering challenge. That was really my first 5 years to kind of re-engineer the whole device to really achieve the goal, kind of like a 40 plus cytokine simultaneously measure in a single cell level, but also you increase the throughput to thousands of single cells. So along the line, we kind of applying the device to sort of single immune cell functional characterization, but also we found you can look at the tumor cells secreted proteins, and turns out it's quite interesting, like leukemia cells or malignant hematopoietic cells in bone marrow can produce those cytokines to hijack non-malignant cells and collectively mess up the whole bone marrow microenvironment. If your therapeutic target in malignant cells only, you don't really achieve the best therapeutic outcome. You have to target both malignant and non-malignant cells. Okay, now, so I'm still working on this, but in very specific context, I got excited about it, I think back to 2015, that's the CAR T immunotherapy. That was the moment when CAR T immunotherapy was sort of wider, sort of applied in a sort of different, both industry and academic clinical trials. And if you look at the basic mechanism of action, how CAR T cell works, and I think Xiaolei is an expert here. And so once the antigen recognized by the CAR, and it's going to trigger a signal transduction through the transmembrane domain and the intracellular signal signaling cascade. And the outcome is the T cell, the CAR T cell become activated to produce cytokines, chemokines, and we really don't know what they're going to produce. We know they need to produce porphyrin to poke the hole on the surface of the CAR T on the tumor cells, and then granzyme produce to get into the tumor cell to kill. But there are probably many other cytokines, chemokines produced, but we just don't know. And the question, can we measure all those to understand the full functional spectrum of CAR T cells? And we did that, but that was challenging to, kind of to do that by ourselves. And interestingly, I think the industry got very excited about our technology. This is the collaboration with Kai Pharma. They took the technology to look at the CAR T cells that they manufactured in their entire ZUMA-1 trial. So this is very kind of first data set on like a 20-ish patients, but eventually they applied this to their entire ZUMA-1 trial, kind of 120 to 150 patients, and then later almost all the patients they tried using their CD28 CAR T cells. If you look at the very first data set they gather, and so all the kind of non-responders versus responders, and most of the non-responders show lower capability to produce multiple cytokines per cell. And you mathematically define something called a polyfunctional strength index. The between responders and non-responders, you do see statistical difference between these two groups based on this polyfunctional strength index, which is the combination of the amount of proteins produced, the amount of cytokines produced per cell, but also the diversity of the cytokines produced per cell. If you look at many other things, I believe you guys know better as an immunologist, you're probably going to do like a gamma interferon production. You're going to look at T cell differentiation states all the way from like a memory to effector, central memory effector, and the naive cells. But none of those really give you statistically significant correlation with the patient clinical response. In this case, there's an initial response, so patient are going to respond or they don't respond. And so, but that was CD20A to treat the non-Hodgkin's lymphoma. So we also had a chance to work with experts at Penn, in particular Carl June and Steve Grubb's group, to study sort of CAR T cells for the treatment of leukemia, in this case, ALL, CL. And if you look at this kind of patient response rate across many different clinical trials, and for AL, the initial response rate is very good. It's called a CR, but actually the CR here is defined as the initial response rate. But after a couple of years, many patients relapse, and it could be up to like almost 50% of patients relapse. So I still remember when I talked to Steve Grubb, so if the kids they relapse, what happened? What are they gonna do? And he was pretty excited about so telling me other treatment regimens that have developed, maybe like a CD19, CD20, kind of dual targeting CAR T cells. But afterwards, he was like 10 seconds of silence, and he said, actually, most of the kids die. So once they develop relapse, right? So now that's still a huge challenge in the CAR T immunotherapy, even for the condition AL, pediatric AL, the patient's response rate could be as high as 90%. So now we decided to look into the relapse versus long-term remission. So such a great privilege to work with Carl June and Steve Grubb. We got all the samples from their very first two clinical trial studies, and total of 82 patients, I believe. So we separate them into different response groups. There's the kind of long-term response over 5 or even 8 years, and the patient relapse within a year, 2, 3. And so because the second trial was initiated around like 2015, so some of the patients have not achieved like a 5-year bar, but they stay in remission. So we still don't fully understand. We still don't know whether or not they're going to stay full remission. So we call them remission. So now we take all those patient samples and we perform single cell multi-omics sequencing, but also we look at their so phenotype based on intracellular, based on the flow cytometry, but we also look at their cytokine production using intracellular cytokine flow, and also using our single cell protein secretion device I just described. But just in this case, now we do a lot of samples. We're using a commercialized device rather than our academic prototype. So totally we sequenced about like 1 million cells, and from 82 patients and 6 healthy donors. And so we look at not just the manufactured CAR T cells, but we also look at how the CAR T cells respond to antigen-specific stimulation. So that's the most important data. So you'll use antigen presenting cells in to stimulate CAR T cells. It kind of mimic what those CAR T cells gonna do post-infusion, at least in the first couple of days. And so you're really looking at the function of those CAR T cells. So this is what we got from this large data set. Total about 1 million cells. We filled out some lower quality data points. We still have about 0.7 million single cells. And so now when we look at all the cells, including stimulated and unstimulated, okay, in the different conditions, it's pretty clear. I think stimulated cells form this cluster, and the basal unstimulated in a separate cluster. And now you can also look at the different patient groups. I think CR and a relapse and a healthy donor. You don't really see that much batch effect, but you do see the sort of very clear separation between stimulated and unstimulated sort of the basal cell state. And so now we try to look into this very rich data set to look for potential correlate with the long-term remission, right? So now we know the group of patients called so BCLL, and that's a long-term 5 to 8 year remission. We try to look for, okay, within that group, any specific subset of T cells there really give rise to the long-term remission. We look for typical what people look at most often, like gamma interferon and TNF, those type 1 cytokines. But we notice this small group of cells called they're producing type 2 cytokines. No matter CD4, CD8, we always can identify that small group of cells. And turns out that small group of cells show statistic difference between long-term remission, long flyers versus the relapse. But the type 1 cells, not really. So that's consistent with the observation in the past of all the CAR T manufactured CAR T cells are very potent. The patient are responding, but whether or not the patient are gonna stay remission after 5 years, turns out that's not really determined by how potent they are, really determined by whether or not they have this sort of type 2 cytokine producing cells in the initial manufactured CAR T cell product. So that was a total surprise to us. But this is what we found from this huge kind of large volume single cell multi-omics data set. And we did some validation using the flow cytometry, but also using our single cell protein secretion. Both assays confirm indeed in the long-term flyers, in the long flyers, so BCL long BCAL group, you do see sort of the CAR T cells producing the type 2 cytokines. I think that they are much more abundant in a long flyer patient CAR T cells manufactured in the beginning. So this is kind of like a crazy, I feel. So I always thought that the long-term response, relapse or complete remission over 5 years, that should not have anything to do with the initial CAR T cells. There must be something within the host. That's when the patient gradually develop resistance. But it turns out, yeah, actually the cells manufactured, you infuse to the patient on day 1 has the crystal ball, all right, to predict what they're gonna do after 8 years. That's pretty unbelievable. And then we try to look for the mechanism. We found so we use the ligand receptor interaction analysis to look for what cells are interacting with that type 2 subset. And so on a cell interaction on the different clusters, we can identify the interaction pairs. And interesting, we saw the IL-2, we still don't fully understand it, but it's interesting. But all others are pretty much sort of the type 2 cytokine and their interaction, sort of their receptors. And then we look for what cells are expressing those receptors. And it turns out that's a group of TIM3 positive kind of dysfunctional exhausted T cells within the whole population of manufactured CAR T cells. I think the hypothesis here is your CAR T infusion product is always very heterogeneous, probably necessary to be heterogeneous. Okay, if you make a pure type 1 positive CAR T cells or pure type 2 positive CAR T cells, you don't get a kind of long-term good response in patients. You need that heterogeneous population to start with. But within that population, you'll always have a group of cells expressing TIM3. And if you don't keep those cells in balance or under control, they're going to it becomes like a drain. So you constantly have more and more cells becoming exhausted and dysfunctional. But once you have type 2 cells, there are sort of the type 2 cells kind of guard the drain, to prevent the further exhaustion or further sort of draining of the whole population towards that exhaustion state. That's kind of my understanding or hypothesis. And then we look into so of the same so different pathways associated with the type 2 cells, but also type 2 regular TIM3 positive cells. And so this is kind of a little bit summary right here. And so you see the pathways associated with the CAR cells, pathways associated with type 1 CAR cells, type 2 CAR cells, and the pathway associated with TIM3 positive CAR cells. You see what have changed once they interact. For example, here it's kind of exhausted or exhausting CAR T cells within the product. Once you have this interaction, you can really boost the cytotoxicity. And no, you don't boost the cytotoxicity. You cannot maintain the balance of cytotoxicity and prevent exhaustion. And so that's what those cytokines produced by the type 2 cells to act upon those TIM3 cells, and the end outcome is the whole population stay in balance. So now the implication is, okay, if we can make the CAR T product with the reasonably good amount of type 2 cells there, and that might kind of indicate some therapeutic strategy, right? But I don't think we can go ahead and test that in patients, but we can do some animal work. And so in collaboration with Li Tang and the EPFL, and so we send the cells with kind of type 2 cytokine activity high and the type 2 cytokine activity low. And still, the population is still heterogeneous. But we have kind of one set of CAR T cells. We did a flow, we did some characterization. We know, okay, that population has relatively high type 2 functionality, and the other population relatively low type 2 functionality. We sort out from the same manufacturer CAR T cells. And then the infusion to the leukemia xenograft mice. Okay, without treatment, in a week, I think the mice develop leukemia and die. And no matter type 2 low or type 2 high CAR T cells you infuse into the mice, you're going to eradicate the tumor cells that worked out. That's exactly what we see in our patients. So pretty much all patients respond as long as you manufacture kind of good CAR T cells. But we did a re-challenge. So after 17 days, and we kind of inject more leukemic cells to the mice, but without adding more CAR T cells. We just say whether or not the mice can reject the leukemia cell again. So it turns out this is a huge difference. Okay, if the mice were treated with type 2 high CAR T cells, I think the animal can continuously reject the leukemic cells. And I think this experiment was done a couple months ago, and already a dramatic difference. And recently we did another challenge, kind of re-challenge twice, and those mice still can reject the leukemic cells we inject, we re-challenge. So in this case, sort of the re-challenge mimics the recurrence, mimics the relapse. So the conclusion is here, those type 2 high CAR T cell or CAR T cell population or infusion product does make difference to maintain long-term remission, even to prevent the relapse. So we also characterize those cells. I think the post-infusion in animal models, and also characterize so many sort of the T cell markers associated with the exhaustion, associated with memory, and it seems like indeed the type 2 cell treated mice, you do see even post-infusion, you see statistically higher level of kind of persistent memory cells and a lower level of those exhausting cells. What about patients? And thanks to Steve Grubb and when the designing clinical trials literally 10 years ago, and the intentionally collect the patient blood samples post the infusion, and those samples are still there. And we say, okay, can we get those patient blood samples to look for the type 2 cytokines in their blood, the level relatively low, but we can still detect. We're looking at the first 2 weeks. Even in the first 2 weeks, you can see statistical higher level of type 2 cytokines in those patients post the treatment, and those patients eventually become a long flyer. So this is statistically significant in this long flyers. And in the second clinical trial, so those patients now approaching the 5 to 8 year bar, but you still see some difference. I think after a week, you do see the sort of the overall the type 2 cytokine signature is higher in the patients who still remain remission until now, and as compared to the patients who develop relapse within the first 2 years. And so I think that's a very interesting result. And also in cancer immunotherapy, I think a type 2 function has now been well appreciated or well recognized. And but in my lab, we observe so many years ago, actually the 3rd generation CAR T cells can simultaneously produce type 1 and type 2 cytokines. And we found that's very rare, very rare. And then we decided to look into type 2 cytokines since then, almost 5 years ago. So now I think we have much better understanding, not just the type 2 function is important in cellular immunotherapy, but we also have much better understanding about the mechanism. What are the recipient cells? What's the mechanism for those type 2, very small subset of the type 2 cells in the infusion product to maintain the homeostatic state of the whole population? And so now next question is, once we know that, how to improve our immunotherapy in the future? And what has not been studied and is the host response. I do believe the evolution of the tumor in patients definitely plays a role, and which we have not looked into at all. But what we have confirmed is, okay, this is not just the host response. Actually, the drug from the very beginning already shows some correlation. So can you do better in your drug manufacturing? That's kind of what we are able to tell. But I definitely believe biology of the host response is so important. We still need to study that in the future. So in animal, probably that's much more durable. But fortunately, I think our collaborators also collected some bone marrow samples from CAR-treated patients. We plan to look into those specimens to understand what happens post-infusion. All right, so back to this slide that really inspired me to develop the single cell and tools to look at the tumor microenvironment, to look at the cellular functional heterogeneity. But as I said, there are 2 questions there, right? Single cell resolution, number 2, spatial context. So far, I think on sort of either technologies or data I've shown has not been able to address the question number 2. But over the past couple of years, there's a really kind of the explosion of the spatial technologies. And so I've been using this chart over and over. In the early days, I call this spatial omics technology landscape, but now I really call this spatial technology family, because oftentimes you need to use multiple technologies together to understand what you want to study. So broadly speaking, there are 2 directions or 2 lines of technology development. One is you can image over and over, and also use some combinatory barcoding trick to increase the number of targeted molecules you want to detect. You want to spatially map down either transcripts or proteins. For proteins, I think many commercialized tools already out there. For transcripts, and also there's a kind of high-plex single molecule FISH, and there's two technologies even demonstrate you can image up to entire transcriptome. So what we have been interested in is larger than the second approach. So can you use sequencing, in particular, I think in a high throughput NGS sequencing to do spatial resolve and a genome-wide mapping of either gene expression and potential other things. Sorry about my voice. I don't know why I cannot advance my, all right, okay. So I really don't think I need to kind of educate the whole community how the technology started, since this landmark paper published by Joachim Lamberg back to 2016 to use the DNA marker rate to capture messenger RNAs, and then you can spatially barcode the messenger RNAs and the transcripts, and then go back to know where they are coming from. Since then, many technologies develop along the same line as the same fundamental principle. So barcode solid surface for messenger RNA capture and the spatial transcriptomics. So what we came up with kind of total opposite. Instead of capturing, we send the spatial address codes into the tissue, and then we can barcode not just the transcripts, we can barcode proteins and potentially many other things, including epigenome, and you can also combine the different omics for spatial co-profiling. And so it turns out it's a very versatile approach to do spatial resolve omics analysis using high throughput sequencing. So when after we published this paper, I think many groups also kind of try to adapt and try to adopt and further tweak. And as far as I know, at least 4 or 5 publications have been out there over the past 2 years using very, very similar technologies as we develop. And so I think 2021, when Nature Methods choose the spatial transcriptomics as the method a year, and then they also highlight our paper in their special issue. They say, okay, multi-omics sequencing goes spatial. So I think that was a nice demonstration. I think in a Cell paper, we did about like dozens of proteins plus whole transcriptome. But the question is, can you really look at kind of hundreds or thousands of proteins? Because from the proteomics folks, if you look at dozens of proteins, that's now considered proteome. But I know nowadays, at least my own experience, if you are able to look at dozens of proteins, you do get a pretty good systems level understanding of the protein signature and a single cell level and a tissue scale. But still, I try to challenge my postdocs. Can you go ahead and do kind of hundreds or even thousands of proteins? It's extremely challenging for academic laboratory to develop thousands, but can we do 100? That worked down, and for both human and a variety of mouse tissues, we're able to do about 300 proteins and a whole transcriptome, and from the transcriptome and from the proteins, when we perform the sort of independent unsupervised clustering, you can see the spatial clusters or spatial tissue features identified by either modality give you very, very consistent results, strong concordance between the two different modalities. The second is beyond the transcriptomics and maybe a panel of proteins. So nowadays, the spatial transcriptomics the whole field of spatial omics is still largely limited to gene expression, and some people are doing proteins. But we were very interested in the underlying mechanism and epigenetic level, what controls the gene expression. So we started to develop this spatial epigenome sequencing. First of all, the accessibility of the chromatin, and using the TM5 transposase called a kind of ataxic chemistry, which is very simple. You use the TM5 transposase pre-assembled with DNA oligo to process the tissue section, and the DNA oligo going to be inserted into the open chromatin region, and afterwards, so afterwards, in our case, okay, we apply our microfluidic device to add a spatial barcode A and then spatial barcodes B. And sort of every experiment you do, you are actually doing thousands of ataxic experiments, and each corresponds to one of the pixels on a tissue. Then you can map down the entire tissue section, pixel by pixel. And if you aggregate, for example, all the pixels from embryonic mouse liver, and you produce this pseudo bulk embryonic mouse liver ATAC data trace, you see pretty strong concordance between our data and the encoded reference data. So recently we try to apply the spatial ATAC to the mapping of human lymph nodes in both normal and malignant, and also in aging lymph nodes, and try to integrate with the single cell transcriptome, so you can really identify all major cell types and see where they are. So this is kind of small region of the lymph nodes. You can using this integrated co-embedding, you can identify all major cell types and go back to see where they are. I think beyond the global accessibility of the chromatin state, can we look into specific histone modifications? So we demonstrate that worked out quite well, still using TM5, but before you apply the protein A TM5, you use the histone mark specific antibodies to recognize those histone marks first, and then use the protein A TM5 to kind of cleave specifically in the DNA loci at a histone modification region. And so in this case, we demonstrate K27 trimethyl, K4 trimethyl, and a K27 acetylation spatial cut tag or spatial histone modification sequencing across several sections of the mouse embryo. And so even without doing this multiplex cut and tag, you just compare data across the several sections, you can see how different histone marks control the development of different tissue or cell types in specific region, right? For example, you see a lot of granular tissue types right here controlled by this particular histone mark, but not really by this one. So now you kind of, in the past, you really need a multiplex cut and tag or multiplex the chipseq to know this. So now even from the serial section, you can get a pretty good understanding of this kind of multivalency, or how the different histone marks work together to control the tissue and the cell development in the mouse embryo. And so we were also able to combine transcriptome and the epigenome. What you see here is the cut and tag and the whole transcriptome co-profiling across 3 different histone marks. And I believe 2 of those are really adjacent. Yeah, these 2 are really adjacent tissue sections from the same mouse brain. And so we didn't do this intentionally as a replicate, but now you look at this 2, both of them, we obtain data from spatial transcriptomics. So now you can look at how consistent, highly consistent between these 2 experiments in terms of the RNA portion of the data. So it's kind of like a replicate that also gives you the confidence how reliable, how robust this assay is. So now next, maybe 5 to 10 minutes, I really want to highlight what's next in my laboratory, and to further develop those tools to understand sort of the immunology or related organs and malignancies. So first of all, I think we still think, the ability to map down either RNA or epigenome from the clinical archival tissue specimens like FFP has huge value. And recently we developed this PathoDB protocol to map down the RNA spatial across all different sample types, but they're all FFP. So now you can see very nice spatial clusters from this is FFP samples, but when you look at the data quality, so that's pretty amazing. And so, for example, here, those are the data we generated, and compare the data quality from the FFP samples and to the fresh frozen samples, the same tissue types, and also the FFP samples done with text vision. But text vision FFP is not a base by base sequence, and that's basically a fish, but you detect the fish probes using NG sequence. So in that case, so I think it's no way to get a better data than that. But turns out we're able to detect even more genes and even more transcripts as compared to that probe base, the spatial transcriptome sequencing, even though our spot size is actually slightly smaller. But what is even more exciting in this case is you are no longer limited to gene expression. So nowadays, the entire field of spatial transcriptomics, I would say, in other words, that's equivalent to spatial gene expression. Not much more you can learn from that data, but RNA molecules have extremely interesting and a rich life cycle. Can you really look at how the RNA molecules, I think, are being transcribed and then processed through all the maturation steps, on a kind of splicing process to give rise to different isoforms, and then how they are translated. So this is very, very rich and interesting biology. And so we are trying to see whether now we can see some of those. So here, this is splicing. Okay, different splicing events. We try to see whether now we can see different splicing events in our data. So turns out we were able to see a lot of those, and based on the different regions, different brain regions, now we can look into kind of region-specific splicing isoform patterns. This is sort of the quantitative analysis of different splicing events in different brain regions. And just to give you two examples, maybe just one, and this particular gene, and you see the expression in isocortex, in midbrain, and also in the hippocampus right here. But when you look at the different isoforms, and the different isoforms show region-specific expression. In particular, this skipped isoform, exclusive expressed only in the dentate gyrus of the hippocampus, but not everywhere else. So this is the first time actually in the FFP samples, you can directly visualize so many different isoforms and to really understand the splicing, RNA splicing biology. And so another exciting and clinically important application is the mutations, right? So I think people are able to do copy number variation analysis using spatial transcriptomics, but for mutations, that's very, very challenging, no matter for single cell or for spatial. But turns out the data quality is so good. I think from our PathoDB, even from FFPE, we're able to do kind of genome-wide single nucleotide variant calling across all kind of 23 pairs of chromosomes, and then you can compute globally and the accumulation of those SNVs, including both somatic and germline mutations. So now you can visualize where you have the highest mutation burden, which is very likely corresponding to malignant tumor cells. And in other words, this kind of like a tumor mutational burden. So you can perform spatially resolved tumor mutational burden analysis. I feel that's kind of totally mind-blowing. In the past, I think we all know the tumor mutational burden is kind of FDA clear clinical biomarker to predict the immunotherapy. Now you can see the tumor mutational burden directly in the tissue in the spatially mapped, spatially resolved manner. And also we took all the mutations to perform unsupervised clustering. Even in the tumor region right here, you can see slightly different subclusters potentially corresponding to different tumor subclones or evolving subclones. So now you can identify using this genome-wide SNV calling. So another interesting aspect is the protein, the RNA molecules that do not really encode the gene, the proteins, but play very important regulatory mechanisms. And for example, the microRNAs. And we actually did a very similar experiment using fresh frozen samples. Turns out the data just not that good at all. And somehow the FAPE tissue processing kind of preserve those very short RNA, in particular the microRNAs, so well, and we're able to detect, for example, from these lymphoma samples, over 1,800 microRNAs, and each microRNA you really can look at carefully using genome browser to confirm, okay, that is exactly the microRNA you want to see. So if you study microRNA, this is almost approaching the entire pool of the microRNAs for human. And so now with that, you can look into microRNAs and their targeted genes and how they are distributed spatially, and potentially can predict the how the microRNA regulate their targeted genes, and or vice versa. And so, all right, so that is sort of what's next, the number one. So can we really understand the rich biology of RNA molecules in the clinical archival FFP tissue specimens? So in the second, I try to be sure, but I really want to highlight the ability to really combine imaging. I know we are burning cache every day in my lab, Illumina sequencer, but the imaging data is so powerful, but can we combine both? So this is the 49 plex. Now we do kind of 53 plex immunofluorescent imaging of human lymph nodes. You can see all different regions, the kind of germinal centers, the follicles, and even the stromal vasculature, and so now we are kind of collecting data from a large number of those lymph nodes to try to build a human lymph node atlas in kind of normal lymph nodes, but also in aging lymph nodes. This kind of ongoing effort. I think eventually, I hope there's going to be a huge resource. But every sample will also have a whole transcriptome, and some of those we have spatial epigenome data as well. But what is very cool in terms of technologies, from the same tissue specimen, we're able to perform spatial transcriptome. So now, sort of every single spot, okay, unlike the 10x Visum data analysis, you can do sort of cell type deconvolution or decomposition, and eventually generate a lot of pie charts. Okay, every spot you have a pie chart to say, okay, certain percentage of cell type A, certain percentage of cell type B. But in our case, we directly visualize every single spot, what you have in every spot, right? This is a relatively large spot, but for some of the relatively small spots, literally 2 or 3 cells per spot, you can directly see what you have based on the immunofluorescence imaging data. So now we try to kind of integrate a spatial high-plex immunofluorescent imaging and a spatial transcriptome to construct this, not just a normal lymph nodes, but also the lymph node, sort of during the transformation from early stage kind of indolent to more malignant lymphoma and try to build this kind of lymphoma genesis spatial temporal atlas. This is just a very early preliminary data to show, okay, from normal lymph node to the lymphoma at different stages, you can build this atlas by combining the codex spatial transcriptome, and also you can predict the cell transcriptome between the gaps to construct this truly single cell spatial multi-omics atlas map. And also with the data from different stages of the lymphoma compared to normal, you can study sort of the shoulder time, how different cells gradually evolve. I think in this case, we just look at the T cells. You do see where the T cells in normal, and when they become sort of in a lymphoma tumor microenvironment, it's a B cell lymphoma. Okay, T cells are not malignant, but you also see how T cells being affected and gradually change their phenotype. And that has been hypothesized, sort of the T cells somehow create a niche to help the B cell lymphoma to progress, to transform. And so another exciting aspect of this combining imaging and the spatial sequencing is, and potential also spatial multiplex immunofluorescence imaging is, and can we add another layer of functional radar, in particular metabolic state? So for in the interest of time, but also interest of the group here in immunobiology. So we know in the germinal center of the lymph node, and in a dark zone and a light zone, you do see sort of the metabolic shift from oxidative phosphorylation to this glycolysis or glycolytic activity high state once the B cells exit the dark zone, enter the light zone, right? And then we try to see whether or not we can see this difference, the shift from oxphos to glycolysis, but cell by cell. Okay, not just globally, we say, okay, dark zone versus light zone, maybe you have more B cells showing higher level of glycolysis. And so now in this case, all right, in the colorism, right? So in this case, we did a multiplex immunofluorescence imaging. And so this is the dark zone, this is a light zone from the same tissue section in collaboration with Dr. Lian Xi at UCSD. We were able to look at the redox activity, which to some degree corresponds to the relative ratio between glycolysis and oxphos. And so now you see, okay, that's right. You do see higher glycolytic activity in a light zone, but that's not a homogeneous. Okay, you actually see quite a few hotspots of those high glycolytic activity cells. But in general, that trend is still right. You do see higher glycolytic activity in a light zone versus dark zone. So we are doing kind of cell by cell segmentation and to find out. So every single cell now you identify by this some multiplex immunofluorescence imaging. Every cell now you can add another layer of critical information, which is the metabolic activity. And so I just show you glycolysis versus oxphos, but you can look at many other metabolic activity using super resolution SRS. But I also want to emphasize what Dr. Xi has demonstrated. It's not just you get a global metabolic radar, but you can really, because there's optical imaging, you can look into the details and a subcellular or even suborganelle level to understand the mechanism within that cell. But now you know what that cell is, you can also map out that across the very heterogeneous cell population in situ in a complex tissue as shown here. All right, don't think I need to summarize, but I hope that is kind of some inspirational closing slide. I should, sort of in the previous slide, kind of pointing towards what the future would look like. And I would like to thank people in my laboratory and all the amazing collaborators and the funding support. And thank you very much for your attention. Sorry about my voice, and I'm happy to answer questions if time permits.